Thank you very much for the introduction and the invitation. It's wonderful to be back in a warm and sunny Scotland. So what I'd like to do in the next 25 minutes or so is show that height has been at the forefront of discoveries and methodological advances in human complex trait genetics for a very long time, addressing fundamental questions about genetic variation, gene and variant discovery, genetic architecture, and polygenic prediction. Time doesn't permit me also to talk about natural selection and adaptation and the effects of assortative mating or inbreeding, but height has played an important role there too in discoveries. So I start in 1886. This is a paper from Francis Galton, where he used the newly developed statistical method of technique of regression to compare the height of children compared to the average height of their parents, in the mid-parental value. And he noticed this regression towards the mean here, so the points do not fall on the line y equals x, and he noted that this slope of this line was 2/3. And we now know that under some assumption, this slope is actually an estimate of the heritability, which is the proportion of individual differences or variance due to genetic factors. Galton also built or sketched a prototype for a forecaster, so a prediction of height, which predicts height of a son or a daughter as a function of the height of the parents. And for the statistical geneticist in the audience, this must be one of the very first examples of a linear mixed model, because we have here a prediction which has a fixed effect, which is the sex, and it has a random effect, which is family history, with a shrinkage factor, which is the factor of 2/3. If we move on a few decades or so, we can see, or just illustrates here, how easy it is to get large sample sizes for height, because it's easy to measure and a self-report is actually quite accurate. This is a paper from Carl Pearson and Alice Lee, where they now used correlation to quantify the resemblance between first-degree relatives. These are all first-degree relatives. And as you can see, these correlations are all about 0.5, very small standard error, means large sample sizes, and also they observe this correlation between spouses. And what is really interesting is if you look at contemporary data nowadays, for example in the UK Biobank, these results are actually very similar, so more than 100 years later. In 1900, Mendel's laws were rediscovered, and there was a fierce debate in the literature whether quantitative traits like height, where quantitative traits like height had the same hereditary and evolutionary properties as the trait that Mendel studied in his piece. And it wasn't until 1918 that a now famous paper from R.A. Fisher reconciled these two different views of complex of traits. And this is really a paper that's the foundation of quantitative or complex trait genetics. And it basically showed that if you suppose multiple genetic variants, each behaving in a Mendelian fashion, and then you have environmental variation on top of that, that it could reconcile the correlations between relatives that Galton observed and Pearson observed under Mendelian inheritance. This paper has got an awful lot in it. It, for example, introduces for the first time variance in a statistical sense and an analysis of variance. It introduces effectively what we now use in GWAS, additive effects, when we regress a phenotype on genotype dosage. It defines additive dominance and epistatic variance. It introduces polygenic models, and in fact, the majority of this paper is how to deal with assortative mating. If we now move on and try to summarize in a very simplistic way more than 100 years of data on human height and look at relatives, we would conclude from all these comparisons that the most variation we observe in the population is due to genetic factors. So On the top left here, we have just the phenotypic correlation I've got here summarized for different kinds of first-degree relatives. Here's the Pearson and Lee 1903 data. These others are contemporary data. And all these correlations, as you can see, they're quite similar. These are data from male twins, identical twins on the left-hand side, non-identical on the right-hand side. These are from different relatives looking at the phenotypic correlation as a function of the degree of relatedness, where one are identical twins, half are these guys over here, and a quarter are, for example, individuals and their grandparents or half-siblings or so. And all these correlations, under some assumptions, and I should stress that, suggest that the most phenotypic variation for height, about 80% or so, is genetic. There is also another experimental design which is completely orthogonal to the design of looking at relatives, and that is to look within families and looking at segregation. And for that you need genetic markers. But what's shown here, and this is estimated from genetic markers, that if we look at full SIBs, although they're on average full SIBs share 50% of their alleles identical by descent, there is variation around that because of segregation and linkage. And in fact, it ranges from about 1/3 to 2/3. So what you can do is to say, well, these SIB pairs that happen to have inherited, say, 60% of their alleles identical by descent, are they phenotypically also more similar than these pairs on the left-hand side? This is a very neat design, but it's not very powerful. You need a very large number of sibling pairs. And this is just a teaser. This is the only unpublished results I'm showing, a teaser for a poster that Julia is presenting tomorrow, where she's showing results from data from a number of different biobanks that we had, where we had in total more than 100,000 sibling pairs, and we estimated genetic variation and heritability from that, and concluded, again, completely consistent with the pedigree information, an estimate of about 80%. Of course, we're nowadays in the era where we want to dissect genetic variation into the contribution from individual loci. And this is a reminder that the contribution of a particular segregating variant to variation in the population and heritability is a function of the heterozygosity under Hardy-Weinberg proportions, where P is, say, the minor allele frequency, and the effect size. And this is the effect size that was introduced by Fisher in 1918. So points on this, all the points on this curve here explain exactly the same amount of variation in the population and contribute the same to heritability, which is rare variants with large effects versus common variants with small effects. And indeed, for height, there have been examples of that where there's points on either extreme. So, for example, it's a long time known, and many of you in the audience know a lot more about this than I do, but rare variants in the fibrillin-1 gene cause Marfan syndrome, and one of the phenotypes of Marfan syndrome is that individuals are on average about 10 or 12 centimeters taller than average. But these mutations are very rare, but the effect size is obviously large. A SNP in this gene here was one of the first, or maybe the first, discovered through GWAS. It's very common in the population, minor allele frequency of 35%, but the effect size is tiny, a few millimeters or so. So when the sample sizes for GWAS were sufficiently large, we wanted to address the question, how much variation is actually captured by all these genotyped variants together without doing an hypothesis test? And that was an interesting exercise, because it turns out that for height, about 45% of phenotypic variation is actually captured by a SNP array with common SNP genotypes. Subsequently, we looked at, well, can we recover additional information by using imputation to a fully sequenced reference panel? And indeed, the estimate goes up from 45% to just over 55% or so. And of course, the next obvious question is, well, because we know that these technologies still don't capture all of the genetic variation in the human genome, what happens if we look at whole genome sequence data? So we tried to address that in a paper from a few years ago, where essentially the question we asked whether pedigree estimated heritability can be recovered or from or reconciled with whole genome sequence data from samples in the population, so not from relatives. And without going into great detail, if you just concentrate on the right side here or on these numbers here, we had data in a collaboration with the TopMed consortium, where we had access to data from more than 20,000 whole genome sequences with a phenotype of Hyde. And we estimated a heritability of 70%, but as you can see, the standard error of that was still large. So we can't conclusively answer this question over here. Fortunately, there are now other resources coming online and are available, for example, the UK Biobank, where we can address the same question for different traits and also for Hyde with a lot more precision. What is also clear, not just from the paper on whole genome sequence data, but also from SNP array data, is there's actually a lot of variation that's explained explained by low frequency variants or rare variants. So what we did here in the 22 paper was looking at the cumulative contribution of genetic variation as a function of the minor allele frequency of the sequence variants. And if there's no relationship between effect size and frequency, then we would expect the genetic variation to go on this line, this diagonal line over here. But what instead we found, and it's been found and reported by many others in the past, is a significant deviation from that. For example, if you just concentrate here, that all these sequence variants, those with a frequency, minor allele frequency of 10% or less, nearly explained 50% of the genetic variation among those sequence data. And again, UK Biobank data or just larger sample sizes will be able to refine this a lot better, because our standard errors when we went to very rare variant spectrum, our standard errors were quite large. So the next question is then coming back to GWAS. And here we wanted to address the question, what happens if you keep increasing your experimental sample size in GWAS? What happens to the discovery element? How many more variants do we discover that are statistically significant? How much variation or variance do they explain cumulatively and together, jointly? And how well do they predict phenotypes in new samples? And both within and between different populations or populations of different ancestries. And this is work from the GIANT consortium and led by Loic Yengo. So the first thing on the discovery, when you have more than these samples were, we had a sample size of more than 5 million. First of all, looking at the discovery, we discovered in total about 12,000 genome-wide significant loads that are jointly statistically significant at a very stringent threshold level. The effect sizes are tiny. The median effect size is less than 1 millimeter. You might be familiar with Manhattan plots. This is not a Manhattan plot. This is actually a kind of density plot where we looked at the overlap of genome-wide significant variants, whether they're within about 100 kb of each other or so. The background of that density is 2, but there are regions in the genome, for example, at the ACAN locus here, where there's more than 20 variants that are all clustered and overlapping. You need all of them in the model to explain variation. We also looked at genetic architecture in this paper, and again, this has been reported before by multiple groups, where you look at minor allele frequency here on the x-axis, on the y-axis, the effect size, and what you see is variants with larger effects, these over here, are tend to be at lower frequency. Consistent what I've shown you from the whole genome sequence data, this is consistent with negative selection. So loci that have large effects have some unknown detrimental effect on fitness and are kept in lower frequency by natural selection. We were very interested in this study to look at aspects of genetic variation in the different ancestries. We had, the GIANT consortium had collected data, not from, the majority was from European ancestry, but it was also data from other ancestry, Hispanic, East Asian, African, and South Asian. So what we did here in this plot here, and I'll go through it in slightly more detail, is look at the correlation of the effect sizes that were ascertained in a particular ancestry, and then looking what the correlation in effect size is when you do a lookup in another ancestry, and both of them adjusted for sampling variation. So all these four graphs here have on the x-axis the SNP effects in European GWAS, European ancestry GWAS, and on the y-axis is the lookup in a different ancestry, and they're color-coded by where the SNPs were discovered in terms of the ascertainment. So, for example, top left one here, SNP effects in European, SNP effect in samples, Hispanic samples, and the correlations are all extremely high, as you can see, from 0.93 to 0.99 or so. The lowest correlation we observed was when the ascertainment was in European samples, and we did a lookup in the African samples. There the correlation, and that's over here, the correlation was 0.74. But I have to point out, although all these results I'm showing here are from common variants, about 1 million common variants, that these are likely to be underestimates of the correlation between whatever the truly causative polymorphisms are. We also wanted to address the question about variation that we hadn't picked up from this GWAS, even though our sample size was 5 million. So what we did, we partitioned all of the 1 million SNPs in the 12,000 genome-wide significant variants, plus or minus 35 kb, and the rest of the genome, and said, okay, where is the variation? And what we found is that in European samples, all of the variation that was captured together by these 1 million SNPs was contained in these 12,000 genome-wide significant variants plus or minus 35 kb, and more than 90% of the variation in the other ancestries. And the reason why this is actually quite interesting is when you look at, how much of the genome is covered by these 12,000 variants plus or minus 35 kb, it's actually only 21% of the genome. So I had anticipated that the whole genome would be covered by these 12,000 loci, but clearly that is not the case. And that's shown on the right-hand side as well. So the next few slides are then turning to polygenic prediction. And the first thing we wanted to do is to quantify how much these 12,000 variants, or actually I'm talking about 10,000 variants now, because these analyses were done in European ancestry samples only, and we discovered about 10,000 SNPs in the European discovery of the GIANT consortium. So the first contrast is between making a prediction from a phenotype on a relative, as in Galton, 1886, versus a SNP-based predictor. So when we look at how well a first-degree relative can predict your phenotype, and that's expressed as prediction accuracy as the variance explained, that's about 25%, which means that the correlation is about 0.5. If we looked at our genome-wide significant variants, in out-of-sample prediction, they explain 40% of the phenotypic variance. This is quite remarkable when you think about it. It's only 10,000 SNPs explained 40% of the variance, whereas it was only in 2010 or so that we needed 500,000 SNPs and saying this is the amount of variation that's captured by all of them. So from 500,000 SNPs, the signal is now contained in 10,000 or so. But as we can see, having a phenotype on your parents, both your parents, is still more accurate. But if you combine them, you do better still. However, we know that using genome-wide significant effects, or just genome-wide significant SNPs for prediction, is not the most efficient thing to do. It's best statistically to use all the information available. And there are a whole range of fancy statistical methods to get better predictors. So we used one of those called S-base-R, and indeed what we show is if you do that instead of going from 40%, these are two samples that we looked at for out-of-sample prediction, one from the UK and one from the Netherlands, and in both cases the genome-wide predictors, genome-wide significant SNPs explained 40% of the variance, whereas the Bayesian one picked up another 5% of variation explained. And the same happens for other ancestries as well. So there are two things to note here. The red bars are always greater than the blue bars, which means using all the information efficiently in a statistical model is better than just picking the genome-wide significant effects. But also, as reported by many groups in the past, the prediction accuracy in other ancestries is significantly lower than when you look at the ancestry where the variants are discovered. And I don't have time to go into this, but there are a number of good sort of population genetic reasons for that. So the obvious next question is, well, how do we do now compared to our Galtonian prediction? And I'll show you that over here. Now, these 1 million SNPs and the Bayesian regression method actually slightly outperforms the mid-parental value from Galton. So SNPs outdo the family history. And this is exactly what you would expect ultimately if you have sufficient power, because a SNP-based or DNA marker-based predictor can actually capture within family segregation, whereas family history cannot. And I couldn't resist in putting this paper up here from a bunch of my friends and colleagues from 2009, published in the Journal from the Society, who at that time were also contrasting the parental average with what then was available for SNP predictor. And they concluded for highly heritable traits such as height, the Victorian Galton's method will long stay unsurpassed in terms of both discriminative accuracy and cost. Well, I'm not going to comment about the cost, but it's clear that it's only taken 13 years to overtake that, which is quite remarkable, I find. So let's look at prediction in slightly more detail now. If we look at prediction over time. So what I've taken here is 3 snapshots of predictions for height from 2008, 2018, and from the paper in 22. And the way I've parameterized the accuracy or how well the prediction does is to look at the difference in mean phenotype between individuals who are at the very top and the bottom of the genetic prediction, a polygenic risk score. That's shown on this scale here. So in 2018, is a paper from Mike Whedon and colleagues, the difference in achieved phenotypic difference between the tails of the population was about 6 centimeters, and you can translate that into what's the effect size of a polygenic risk score. And at that time, the effect size of a polygenic risk score was about 1 centimeter. So that's 1 standard deviation of the polygenic risk score equates to 1 centimeter in effect size. If you move on to 2018, where now the discovery sample size has gone up to 700,000, the difference between individuals in the top and bottom of the distribution of polygenic scores is now 15 centimeters, which translates in a standard deviation of the polygenic risk score of about 3 centimeters. So the effect size of the prediction has increased due to increased experimental sample size. And if we now go to 5 million, the difference between the top and bottom is 25 centimeters, and 1 standard deviation equates to between 4 and 5 centimeters. Now, this is quite interesting, I find, because we would expect the same to happen in principle when you do this for common disease, if you think about applications in the real world. Finally, a word about prediction. As many of you will know, prediction, it may be very accurate and we might get high correlations, but it's still not diagnosis. And the way I've tried to describe that is on this graph over here, where I have on the x-axis the amount of phenotypic variation, individual differences that are explained by a genetic predictor, and on the y-axis, what the standard deviation in the actual outcome would be around the predicted value. So if you don't know anything, so your genetic predictor doesn't explain anything, you can make a prediction about somebody's height, and that would be the population mean. And the standard deviation around that is just the population standard deviation, which is about 6.5 centimeters for height. If we go to the GWAS from 2018, which explained just under about 20%, just under 25% of variation, that's gone down from 6.5 to about 5.5 or so centimeters. So gone down a bit, but there's still a huge amount of variation in actual outcome given the predicted value. And if we go to the 22 one, it's gone down to 5 centimeters, even though the correlation between outcome and predictor is of the order of 0.7 or so. But still a lot of variation, I'll explain. Finally, hypothetically, imagine that if the heritability of height is really 80%, and we've captured all the contributing segregating variants and estimated their effect size without any error, we would still have a standard deviation of outcome given the predictor of 3 centimeters. And the reason for that is, of course, that we can't predict the environment from a genetic predictor. So I don't have a conclusion slide, but I hope I've convinced you that height has been at the forefront of discoveries and methodological advances in human complex traits, so it's a model trait. And I'd like to finish by showing you now an old slide. This slide is actually more than 5 years old, but it's the only one I have where we tried a normal distribution of height. You can see. And just a few people to highlight here. Jian Yang, who did all the work and led all the work on the SNP heritability and of course, wrote GCTA. Loic at the back here, some over here. Julia, who is showing the poster tomorrow, is over here. Mike Goddard is based in Melbourne, but he's an honorary lab member. And also, I'd like to acknowledge all the wonderful collaborators, many hundreds, probably thousands around the world who've contributed to many of these studies. And for the younger folks in the audience, just read the bits in red. Thank you.
Thank you, Peter. You saved 2 minutes, so we can offer one urgent question, perhaps. Yes?
Peter, hi. Tim Frayling from Exeter over here. Hi, Peter.
Oh, hey, Tim.
Could you elaborate a bit more on the prospects for incorporating rarer variation into polygenic risk scores?
Yes, I'd first like to point out that that SNP I showed, that was one of the first identified from GWAS, was by Tim here, I think. So there are many ways of utilizing the polygenic predictions, for example, for discovery of rare variants, because if you go to a new sample, you can basically take out a chunk of the variation by adjusting for a common variant polygenic risk score. I don't know whether Tim means that. Secondly, there is strong evidence, and one of the things that I know several groups are looking at is whether this concentration of genetic variation around these genome-wide significant loci also applies if you look at rare variation, for example, exome variation or just low-frequency variants. But clearly, polygenic risk score are an incredibly powerful tool for research because you can essentially create a new phenotype in your own smaller sample that gets rid of a whole bunch of variation, which is essentially noise variation if you want to test hypotheses in your own sample.
Thank you.
Thank you. Thank you again to all the speakers and you for the attention.